Good morning, everyone, and the topic of today's lecture is genetic frontiers. And genetic frontiers is about the DNA technology, the genetic modification, and specifically about the genetic modification of the plants. And selective breeding, in which humans breed plants with desired traits, has been used for thousands of years to develop better food crops such as corn, apples, and beans. The first farmer who ever decided which seeds to save for the following year's crop was practicing selective breeding. However, selective breeding requires a long time to produce results and does not always have the desired outcome. Corn with the protein content of soybeans, for example, is beyond the capacity of selective breeding. Using organisms to produce products that benefit humanity is known as biotechnology. Biotechnology does not necessarily refer to technology in the 21st century sense. Selective breeding of plants is an example of biotechnology, and so it is the use of yeast, that is a single-celled fungus, to cause bread to rise during baking. However, advances in genetics promise unprecedented control of inheritance. When people talk about biotechnology today, they are often using the word in a narrower sense, referring to the manipulation of the genetic material in cells so that organisms with new characteristics are constructed. Such modification of the DNA of an organism to produce new characteristics is also known as genetic engineering. So, genetic engineering differs from the selective breeding in several aspects. One difference is that genetic engineering enables new strains of organisms to be developed in a fraction of the time required for selective breeding. For example, this uh, a gene from a common soil bacterium was engineered into cotton, and that gene codes for the production of a protein that acts as a natural insecticide. In a field test, cotton plants were subjected to an insect infestation, and genetically engineered plants received little damage, and as a result of being healthy, produced larger balls of cotton. The untreated plant was weakened by insects and produced much smaller balls. And with traditional breeding methods incorporating genes for disease resistance into a particular crop plot might take 15 years. Genetic engineering has the potential to accomplish the same goal in a fraction of that time. Moreover, genetic engineering differs from traditional breeding methods in that it can make use of desirable genes from any organism, not just those from the species of plant being improved. If a gene for disease resistance in uh, petunias would be beneficial in tomatoes, the genetic engineer can splice the petunia gene into the tomato plant DNA to produce a disease-resistant tomato plant. This process could never be carried out by traditional breeding methods because petunias and tomatoes are different species and cannot interbreed. Recombinant DNA technology is a field of biology that started in the mid-70s. It's actually had its roots in 40s with genetic studies of bacteria and bacteriophages, the viruses that infect them. After decades of basic research and the accumulation of extensive knowledge, recombinant DNA technology became feasible and available to the many scientists who now use this method. Recombinant DNA technology permits the formation of new recombinations of genes by isolating genes from one organism and introducing them into either a similar or an unrelated organism. Using recombinant DNA technology, genetics can insert DNA not only into unicellular bacteria but also into cells of plants and animals. Once inside the cell, this foreign DNA may be expressed. A foreign gene may alter the genetic composition of a bacterium, for example, so that it produces novel proteins such as human growth hormone. Scientists are not the only ones who practice recombinant DNA technology. Organisms have natural mechanisms for changing genes, and such gene transfer have apparently always occurred in nature. For example, bacteria sometimes transfer DNA through a special tube that extends from one cell to another. The transfer of genetic material from one bacterium to another by a bacterial virus is another example of natural genetic exchange. However, these natural experiments have occurred randomly and certainly not to satisfy human goals and desires. Inserting a foreign gene into a cell requires several steps. The gene that is being introduced into a cell must be isolated from the original cell, used to construct a recombinant DNA molecule, or transferred into a new cell identified as having been taken up by the cell and expressed in that cell. We consider each of these steps in detail. To begin the process of genetic engineering, the investigator must isolate the gene of interest. The researcher breaks the cell's DNA into manageable fragments by using bacterial enzymes known as restriction enzymes, which cut DNA molecules at specific base sequences. The restriction enzymes generally recognize a specific DNA sequence 4 to 6 base pairs in length. One restriction enzyme recognizes and cuts DNA whenever it encounters the base sequence, whereas another cuts DNA at the sequence of the uh, G88DCC. So this is like a uh, base sequence, uh, and discovering restriction enzymes was a major breakthrough in developing recombinant DNA technology. Today, many restriction enzymes which with its own characteristics are readily available to researchers. Many base sequences recognized by restriction enzymes read the same from left to right and from right to left. That is, the base sequence of one strand reads the same as the bases on its complementary strand read in the opposite direction. For example, note that the DNA base pair sequence AAGCTT reads the same as the reverse of its complementary sequence TTCGAA. TTCGAA. So, like that. Oh, the mirror of like there is a center here, so this are mirrors. When the particular restriction enzyme recognizes the AAGCTT sequence, it cleaves the double-stranded DNA between A and A on each strand. This action leaves one strand longer than the other on each DNA fragment. The single-stranded stub that extends from the end of each fragment of DNA is called a sticky end. DNA segments from different organisms potentially can recombine with one another if they were broken apart by the same restriction enzyme. Each restriction enzyme always cuts the same base sequence, no matter what kinds of DNA it cleaves. For this reason, the sticky ends of the DNA are complementary and can be joined or spliced by an enzyme known as ligase. Restriction enzymes split the potential donor DNA into many sticky end fragments. The next step is to construct recombinant DNA molecules by combining these isolated DNA fragments with DNA from another source, which acts as a vector or DNA carrier. The vector carries the gene from the organism into the cell of a second organism, where the gene is incorporated into that organism's existing DNA. The most common vector in recombinant DNA technology comes from bacteria. In addition to having one large circular DNA molecule that contains a bacterium's genetic information, some bacteria have small circular DNA molecules known as plasmids that carry one or two genes. So this is called a plasmid. Uh, when a bacterial cell divides, any plasmids that are present within the cell are replicated along with the bacterial chromosome, and the copies of are distributed to daughter cells. When the plasmids are used as the genetic vectors, designed genes are inserted into the isolated plasmids, which are then incorporated into bacterial cells. Each plasmid and its own genes plus the inserted gene are then replicated every time the bacterial cell divides. As a result, the entire bacterial population descended from the original bacterial cells contains the novel genes in its plasmids. To construct recombinant DNA molecules for which a plasmid is the vector, the investigator treats plasmids isolated from bacterial cells with a restriction enzyme. Uh, so this is a plasmid, then treated with a restriction enzyme. Because the restriction enzyme cuts the plasmid at the base sequence it recognizes, the formerly circular plasmid now becomes linear molecules of DNA with sticky ends. These plasmid strands uh, are then mixed with the sticky DNA fragments from the donor cell. So this is the sticky DNA fragments, and then the scientist uses ligase to join the sticky ends. Uh, and then we have a new circular plasmid. The result is a mixture of recombinant plasmids, with each recombinant plasmid containing a different fragment of donor DNA. Although the vast majority of plasmids in the mixture do not contain the desired gene, a few will have exactly what is needed. To identify the plasmid that contains the gene of interest, the investigator amplifies all the plasmids, that is, many copies of each are made so that there will be millions of copies to work with. Amplification takes place inside bacterial cells as they divide. The recombinant DNA molecules now existing as plasmids are then taken into bacterial cells. Under normal conditions, bacteria do not take in plasmids. However, some bacteria receive plasmids under certain conditions, such as when the bacteria are treated with heat and calcium. Bacteria that take in plasmids are said to be transformed. Because only a small portion of the bacteria take up the recombinant plasmids, even under these special conditions, there has to be a way to determine which bacteria contain the genetically altered plasmids. Fortunately, plasmids often carry genetic information that makes the bacterium resistant to certain antibiotics, such as penicillin and tetracycline, that kill or inhibit bacteria or other microorganisms. If the plasmid that is used as a vector has a gene for resistance to a particular antibiotic, then investigators can grow or culture the bacterial population in the presence of that antibiotic. Only bacteria that have taken up the genetically altered plasmids resist the antibiotic and survive. The rest are killed by the antibiotic. So we take the uh, plant DNA, coat it with the instruction enzymes, and then we produce a recombinant DNA, and then they are split with the plasmids that are resistant to the antibiotic, and then they are taken with the bacteria, and then they are planted at the antibiotic medium, and with the plasmid bacteria will survive, and without plasmid it will fail to grow. To select the cells with the desired gene from among all other genetically altered cells, the researcher spreads a sample of the bacteria culture on plates of nutritious agar or a solid growth medium. If the sample is dilute enough, each bacterial cell is separated from other cells on the plate. As each cell reproduces, it gives rise to a colony, a cluster of millions of genetically identical cells. Each bacterium in the colony contains the same recombinant plasmid, because there may be as many as several thousand colonies, each containing a different portion of foreign DNA. Researchers must then identify which colony contains the gene of interest, that is, the one they hope to transfer into a different organism. Because a single gene is only a small part of the total DNA in an organism, isolating the piece of DNA containing that particular gene once the organism's DNA is spread among multiple recombinant plasmids is like finding a needle in a haystack. A needle could be found in a haystack of any size if one had a metal detector. The genetic equivalent of a metal detector can find just the desired DNA-based sequence among the vast number of genes in the bacterium's chromosomes. This device, called a genetic probe, is a radioactively labeled segment of RNA or single-stranded DNA that is complementary to the target gene. For example, suppose we want to identify the gene that codes for the protein nitrogenase, an enzyme involved in nitrogen fixation. Because we know the amino acid sequence of nitrogenase, we can synthesize the macrosomal RNA, matrix RNA that produces it. This matrix RNA, which is the probe nucleic acid, attaches by complementary base pairing to the DNA that contains the that the base sequence needed for production of nitrogenase. Because the synthesized matrix RNA is radioactively labeled, It can be detected by X-ray film. If after this treatment any radioactive DNA is detected in a particular colony of bacterial cells, that is the colony that contains the gene for the production of the desired protein, in this case nitrogenase. Even though a gene is now known to be incorporated into certain bacteria, growing the bacterial strain would not necessarily produce the desired protein. The gene of interest cannot be transcribed and subsequently translated into a protein unless it is associated with a set of regulatory genes that in effect turn the gene on. When these genes are added to plasmids, the bacterial cells can transcribe and translate the gene of interest. The plasmid is replicated and distributed to its daughter cells during cell division. When a recombinant plasmid, one that has foreign DNA spliced into it, replaces in this way, many identical copies of the foreign DNA are made. In other words, the foreign DNA is cloned. Although several vectors for the introduction of genes into plant cells are used, the most widely used vector is the crown gall bacterium, which produces tumors in a variety of plant species. This bacterium causes a disease by inserting DNA from its plasmid into the plant cell. The plasmid DNA goes to the nucleus and is integrated into a plant chromosome. The inserted DNA instructs the plant cell to produce abnormal quantities of plant growth hormones. The result is repeated cell division, forming a tumor. It is possible, however, to disarm the plasmid so that it does not induce tumor formation and to incorporate desirable genes in the plasmid of the bacterium for inserting into a plant host. The plant cells into which such genetically altered plasmids are introduced are essentially normal except for the newly inserted genes. Genes placed in the plant cells in this fashion are transmitted to the next generation by sexual reproduction or asexual reproduction. There are several problems with using the bacterium plasmids to introduce foreign genes into plants. First, only eukaryotes are susceptible to this bacterium. Important monocots such as corn, wheat, and rice are outside the host range of the crown gall bacterium. Viruses are also used to introduce recombinant DNA into cells, and they are commonly used as vectors in mammalian cells, including human cells. These viruses are disabled so that they do not kill the cells they infect. Instead, the viral DNA, as well as any foreign DNA they carry, becomes incorporated into the cell's chromosomes after infection. Other methods used to introduce foreign DNA into a cell do not involve a biological vector, that is, a bacterial plasmid or virus. In some cases, the researcher alters the cells chemically or by electroporation, delivering the electric shock to make the plasma membrane permeable to the DNA molecules. Unfortunately, not all plants take up DNA readily, particularly the cereal grains that are a major food source for humans. One useful approach is a genetic shotgun. Researchers caught microscopic gold or tungsten fragments with DNA and then shoot them into plant cells so that they penetrate the cell walls. The gene gun was first used successfully to introduce genes into algal and yeast cells, but it is now used for plant and animal cells as well. For example, genetics uh, are used such as an approach to transfer a gene for resistance to a bacterial disease into cultivated rice from one of its root relatives. A further problem with the gene insertion in plants is that researchers must use plant tissue culture to regenerate whole plants after individual cells have been genetically transformed. Tissue culture techniques do not work for all plants, although more than 1,000 species have been regenerated in this way, and the number increases yearly. Recombinant DNA techniques cannot be used for plants in which tissue culture is a problem. An additional complication of plant genetic engineering is that some important plant genes are located in the chloroplast instead of the nucleus. Recall from the previous process that chloroplasts are essential in photosynthesis, a process that is the basis of plant productivity. Chloroplast genetic engineering is currently the focus of much research interests. Researchers have many applications for cloned DNA. They may clone a gene to obtain the encoded protein for some industrial or pharmaceutical process. Regardless of the particular application, before they engineer a gene, researchers must know a great deal about the gene and how it functions, the usual first step in determining the sequence of nucleotides. In the 1990s, the advent of automated DNA sequencing machines connected to powerful computers let scientists sequence huge amounts of DNA quickly and reliably. Sequencing machines are powerful and decode about 1.5 million bases in 24 hours period. These advances in sequencing technology made it possible for researchers to study the nucleotide sequences of entire genomes in a wide variety of organisms, both prokaryotic and eukaryotic. Much of this research received its initial impetus from the Human Genome Project, which began in 1990. The sequencing of the 3 billion base pairs of the human genome was essentially completed in 2001. The genomes of more than 100 species have been sequenced since now, and several hundred or more and are in various stages of planning or completion. For example, scientists finished the rice genome in 2005. This was the second plant genome to be sequenced since uh, the model research plant uh, Arabidopsis thaliana was the first. We are in the middle of an extraordinary explosion of gene sequence data, largely due to automated sequencing methods. DNA sequence information is now kept in large computer databases, many of which are accessible through the Internet. Examples are databases maintained by the National Center for Biotechnology Information and by the Human Genome Organization, that is HUGO. Genetics use these databases to compare newly discovered sequences with those already known, to identify genes, and to access many other kinds of information. By searching for DNA sequences in a database, researchers gain insight into the function and structure of the gene's protein products, the evolutionary relationships among genes, and their variability among gene sequences within a population. So I think that is all for today, and thank you for attending the class, and see you next week.